Okay, so coming to today's Q&A session. So this is about, it's actually based on one of our e-learning collections. So what I mean by that is that this e-learning collection is a curated set of EMBL-EBI online courses, which provide you an introduction to chemical biology or related informatics resources. So if you have interest in this topic, if you go to this e-learning collection, you will find several tutorials, recorded webinars as part of this course, and you might find it very, very useful for you. So for today's Q&A session, it was recommended that you go through this e-learning collection before joining the webinar. But if you haven't gone through it, that's still fine. You can still listen to all the questions being asked here, or if you can also ask your question here. All right, and in today's panel, we have Helena, who is an outreach officer in Open Targets, and Annalisa, who is product manager of Open Targets. And we have Harris, who is a chemoinformatician, part of chemical biology services. And we also have Nicholas, who is data mining and analyst scientist at chemical biology services. So yeah, so without further delay, I would hand over to our panel to introduce themselves. So let me first stop sharing my screen. Okay, so can start with the Open Targets team.
Yeah. Hi, so we're just going to share some slides just to very quickly introduce Open Targets to you. So hi, I'm Helena, I'm the outreach officer.
Annalisa, product manager.
And so Open Targets is an industry academic collaboration. So we have a number of partners, and the EBI is one of them. But we have a very broad remit. So here's kind of how we think about the work that we do. So this is the Open Targets knowledge cycle. So at the top, we have public data, of which the EBI resources provide a lot of the data that we use. This data is taken up by our informatics research program and the different projects that we have within that. And the data is aggregated and integrated into a variety of platforms. So the main one that we will focus on today is the Open Targets platform, but we also have one that's focused on genetics data, which is called Open Targets Genetics. But yeah, so the Open Targets platform will take all of this data and will integrate it into one user interface that's easy to navigate, that is systematic, and that kind of presents all this information to you in a way that's hopefully easy to understand and consistent across the different databases. So hopefully using all this data, using the way that we present this data, you can generate some therapeutic hypotheses. And then on the other side of it, we have our experimental program, which will take some of these therapeutic hypotheses, develop them, and try to look into either the biology of different diseases or into trying to find novel targets to try and ameliorate those diseases, and to try and generate new data that can then become public and that can feed into the platform. So these are the two sides of Open Targets, the experimental side and then the informatics side, which is what we're focusing on today. So to give you an idea, just in case you haven't gone through the course, so the Open Targets platform, which you can access using this URL at the bottom, platform.opentargets.org. So it's a data integration and analysis tool, which allows you to systematically identify and prioritize drug targets. It integrates over 20 different public data sources to build and score associations between targets and diseases. In addition to information about targets and diseases, we also have information about drug. So if you were to go to this URL right now, you would be able to search for a target, a disease, or a drug, and to explore all the evidence that we have for all three of those entities. And then this is an open source platform that we create, so the code is available for you to take a look at. There's a variety of different ways in which you can access the data. So we have this UI here, but we also have a GraphQL API. You can use BigQuery, or you can use our data downloads directly and just get the files that we use for the platform. And I think that's everything I wanted to say. Here's just a few key links if you're interested. So social media, if you want to follow us, and then some key links to the platform, the blog where we describe whatever changes we'll make in the platform, and the community and documentation, which are just really good places to get started if you want to understand how to use the platform. But obviously also we're here today if you have questions about how to use the platform. That's what we're here for. So that's just a really quick run through the Open Targets platform, and I will hand over back to Ajay.
All right, Nicholas, you can go next.
Hello, everyone. My name is Nicolas Bosque. I'm part of the ChEMBL team, I've not prepared introduction slide to introduce ChEMBL because we believe that with the question that you have already asked, it will introduce very well ChEMBL, how we get the data, what we do with them, and how you can access them. So just briefly on me, I'm more or less a data scientist. I'm not involved in the data curation or treatment. So I'm kind of a privileged user. I can access the data very easily. I can give feedback to my colleagues, and it's really cool to be able to do that as part of the team. Maybe Aris, you want to say something as well?
Yeah, Haris. I also work for the ChEMBL team. I'm a chem informatician, so basically I work with analyzing compound data and drug data for ChEMBL. And lately, I'm mostly responsible for updating the drug base, which we will introduce you through our slides, which is basically like a platform which is released via ChEMBL, and it basically contains only drug data. And then Ajay.
All right. Thank you. Thanks to our panel for introducing yourself. So for Q&A session, first we'll go through all the questions that have been asked during the registrations. But feel free to type in your questions as all the questions are being answered. So all right. So first question is for ChEMBL team is, where do you get the bioactivity and drug data from?
Okay, so I will answer this question, and in particular, the aspect around biochemical data. So I'm sharing my screen. Okay, good. So this is a brief overview of where the bioactivity data comes from. So ChEMBL has been around for about 13 years now, and we accumulated about 2.3 million compounds with related properties and activities. The core data of the database comes from the scientific literatures, where we extract relevant information from articles that are from different fields, but related to drug development and agronomy. We also get information that are shared with us by PubChem. We extract information from patents when the compound that are protected by the patent are associated with bioactivities. We also have a lot of data and that are related to the toxicity field. And an important aspect of ChEMBL is that other organizations, private and non-for-profit, are able to directly deposit data sets into the database. So it's very convenient for them to do that. You don't have to write necessarily an article to do this. Of course, they go also through the same curation that will be introduced later. The latest aspect of the data in ChEMBL is what we call clinical research component marketing drugs, and that's Aris that is going to give you a flavor of what it is. So Aris, the floor is yours.
Thanks, Nicholas. I'm going also to share my screen.
Or I can just
Share
if you want.
On.
Yeah. Okay.
Am I sharing the right screen? Yeah, I hope so. All right. So as Nicholas said, we have also drug data in ChEMBL. We have around 14,000 drugs in ChEMBL. And this is basically stored in an internal database, which we call it drug base, and it's released via ChEMBL. So you cannot access it like as drug base, but all the data that we store in there, they are you can search them via the ChEMBL website. So in drug base, we take data from that come either from clinical candidates, which is at the very late part of the drug discovery and development process, or we also take data that they come from approved drugs, sources like FDA, for instance, or Orange Book. And besides the drugs that the drug data that we store in ChEMBL, we also store the indications of the data. So here I have a more detailed list of sources that are classified into drug development and approvals. We can see that for drug development, we take data from clinicaltrials.gov, which is our main source of information, because most of the drugs come from clinical trials, also using applications, INN applications, ADC classifications. And for approved drugs, we take data from Orange Book and FDA and also information for warnings that we may find in FDA labels. And you can basically access this data, as I said, when we migrate drug base to ChEMBL. And you can see all the relevant information in their compound cards in ChEMBL, as I show here. So you can see whether a compound, for instance, can be a synthetic compound, whether it's like a chiral compound or not, a prodrug, the way that it's being administered. Oops, sorry. Whether it's marketed or not, whether it has a black warning or not. So there is a lot of information that you can access via ChEMBL. So Ajay.
All right. Okay. Thanks, Nicholas and Harris. I'll stay with ChEMBL team for the next question. The next question is, how do you curate, maintain, and update the data?
Okay, I'm happy to answer also this question. So both for drug base and ChEMBL, it's like a more generic approach. We work with compounds, and in ChEMBL, there is also the target information. So in particular for drug base, there is the name and structure mapping, and also the indications and the warning, the black box warning annotations. For both drug base and ChEMBL, there might be also a part of manual curation of data. So here I'm showing you an example of a using application PDF documents, which USIN stands for United States Adopted Names. So here on the left-hand side, we have like a small molecule, and on the right-hand side is like an antibody. So we have chemistry curators in our group that they manually extract data from these documents. And this is just an example. This applies to GUSENS, but it may apply also to other sources. I'm just showing you an example here. So the structure information comes from these kind of documents, as we see here or here. And also some of the indications might be taken from these GUSEN notebooks as well. Then we also extract the data from full text articles. And we also extract from these articles, apart from compounds, also target data. And our main journals of interest are the ones that I'm showing you here. And all these goes through an extensive curation process in ChEMBL. And once we have extracted all this data, either from literature or from other data sources that I have mentioned before, then they go through this curation process. So for our compounds, we have an established structure pipeline that you can access from GitHub. I'm showing you the link here. So all the compounds that they have been extracted from these documents, they pass through a standardization process. There is a salt stripping for the creation of the parents. And all parents and salts are connected through the molecule hierarchy that is stored in the hierarchy table. And you can also find it through other relative compounds on ChEMBL. There is also a calculation of different properties because there is a very diverse the different sources that we have diverse measurements in ChEMBL, and some of them are quantitative, such as binding affinities, or some of them are qualitative. So we have to also standardize the different types of data, and of course we standardize the different type of units. And there is also a mapping from the assays to targets. So we register the targets onto the relative tables in ChEMBL that you can also find all this information online.
All right, thank you. Thanks, Aris. So now next question is, I'll first go to Open Targets team for this question. Is there any validation or quality control process when you get data to your database and that you take before including it into your database?
Yeah, maybe I can take that one. So I'm not going to use any slides for that, but as Elena showed you in the introductory slides, we have in the knowledge cycle she showed, we integrate publicly available data sets, so data from publicly available resources like ChEMBL or Reactome or Cosmic, for example, into the platform. Now, at that level, because we trust, and we also integrate from publicly available data from papers, right? So at that level of integration, we trust that the resources like ChEMBL, for example, have done all the checks and QCs for us and validation for us. And also from the peer-reviewed paper, we trust that the paper has been peer-reviewed and data is actually accessible for integration. The second layer that we validate is that Open Targets, so data in Open Targets has to conform to a certain schema, which you might look at it as a format, like a POSH way to format the data. And of course, if you think about the amount of data we integrate and score, we need to have a certain harmonization of the data, right? So the validation we do is actually against the schema. If the data that we integrate conforms to the schema, we can use it and score it and use it for target validation. If it doesn't conform to the schema, we cannot use it. So this is just to cut a long story short. This is the main validation step we take in our input pipeline. I don't know if Elena wants to.
No, yeah, that's the
validation bit.
All right, thank you. And I think, Harris, you already, I think, partially answered this question in your slides already, but do you have any other comment to add to it?
No, I'm fine. I think we've covered, from our slides. The only thing I forgot to add during the presentation about the curation, and I think it's a very important thing, just going back a little bit, is that the curation process is an ongoing process. So issues can be fixed either during every release. We may come across with issues that we either identify ourselves or they may be identified from our users. So I'm showing you here our help desk. So we welcome any comments about or any issues about our ChEMBL releases, and we try to address them. And in every release, apart from the new data that it comes with every ChEMBL release, we also try to fix any arising data from previous releases.
That's a very good point. Actually, I want to add a very quick question. So Helena or Annalisa, do you sometimes get like user email or something about they mention some issues that is this validation correct, or I'm looking at this data from some user, but is this correct or not? Do you get some emails like that?
Yeah. I mean, yes, we do. Elena can add. We do get emails, and we also have, because Open Targets is a partnership, we have emails from the partners, from pharma, commenting on our data, but also on the common user finding issues with the data. And as Elena will tell you now, we actually open a community page where an Open Target community page where the users can actually now open issues, and there are threads on data issues. So it's actually very nice for us to follow up discussions in between users.
Yeah, so we no longer have a help desk email address, but we ask people to post their questions into this community page, because then the idea is that it kind of creates this sort of database, that searchable database of help about Open Targets. So we'll type it into the chat box, but it's just community.opentargets.org if you want to take a look at the questions that we've already answered there.
Yeah, that's very good. Okay, so we'll go to the next question, and I think it fits with both Open Targets and ChEMBL team. It's about accessing the data. So is there a way to access the data programmatically? So if there is more computer savvy users or bioinformaticians, how they can access the data programmatically?
We want to start, guys, or shall we kick off? We're going to kick off. We actually have a slide on data access, but yeah, the Open Targets data can be what's going on here?
It's too many tabs.
Yeah. Can you see the accessing platform data?
Yeah.
Yes.
So we have many ways for the users to access our data, and this is, as you said, AJ, we have loads of very heavy programmatics users. So this was very important to us to get very accessible data. So of course the users can download the tables from our user interface, but talking about programmatic access, we have a very nice, it's actually a new re-implemented API, the GraphQL API, where the users can actually use different endpoints to query our data, and everything is reported very nicely, reported in our documentation page. So I really encourage you to go and check all the also example queries you can have and you can perform with our GraphQL API. We also have and deposit all our data onto a Google query, Google BigQuery bucket, so the users can actually also query the bucket with our data on Google and can actually export all the queries. So this is like an extra access point we give to our users. And also,
and just to add on that, that's now accessible through the public data. So actually, you don't need to pay anymore to access our data through Google Cloud, which is really cool.
Good point, yeah. And then, of course, this is classic download, which is less programmatic, but still, for a big download, you do need some piece of code. So all the users can download all the data sets from the FCP and from the Google Cloud. Mm-hmm. And yeah, that's, I think, all from us. Back to you, Ajay.
Okay, thanks. Harris or Nicholas, would like to add?
I was saying, so for ChEMBL, we also have different ways to access the data. Of course, we have the classic UI that anyone can access, and you can use the search field to query any kind of data, compound, targets, assay, or a document. But if you are a bit more techy, then of course you can download a dump of the ChEMBL database. The link are accessible on the main page of the website. You can download the database in different formats. But I really want to focus here on the web services, which are also accessible through the landing page. And so this web services, they allow you to access the same data as if you were using the UI, except that instead of having a nice compound report card, you get something which is also nice, but in a different way, and it's more convenient to access the data through this format in an XML or in a JSON when you need to access a lot of data very efficiently, very quickly. And of course, this you have the URL here that well, you see it's very well explained. I think you've got the stem of the web services, and then you select which type of data you want to access, and then you can type different entity. And so, for instance, for paracetamol, you type the ChEMBL ID of paracetamol, and you will get all the properties of this drug. You have all the documentation related to the web services on our website. The link is on the slide here. And you can see that we have a lot of different resources that allows to access activities, assay, ATC, binding site, and so on. The API can be tested on live and so you don't have to actually write any code. You can just submit a molecule ID, for instance, and you will get the result. But it's usually more interesting to integrate the web services into a program. An important point here is that you cannot download the whole ChEMBL using only one request. For performance reason, we have a pagination of the data. So if your requests return too many information, in practice, if it's more than 20, you will have to download the data page by page. And this is managed through the URL. As I already said, we have different endpoints, and you might wonder which one you need to download the kind of data you are interested in. Hopefully the names of the endpoint are clear enough. So if you want to download a molecule, you will use a compounds one, a target, you will use target 1, and so on. You have the list here. For the sake of time, I will move on a bit quickly here. You also have, again, all the information available in our website. We also expose several functions that can be very useful. Like, so if you want to do a similarity search, if you have a query component and you want to know if there is something similar in ChEMBL, you can use the similarity search. You can also do a document similarity, and also if ChEMBL ID lookup. We mentioned drugs earlier that are an important subset of the ChEMBL data, and so to access them easily, they are also at their dedicated endpoint to find a drug in particular, or the mechanism, the indication, or the drug warnings related to this specific drug. The URL, as I've said already, is formatted in a very specific way, where you have the stem that never changed. Then you say which resource you want to use, which field in the resource you want to search, and then you can add filters, so it can be equal, but it can also be, if it contains certain things. You have to be careful about the formatting of the URL, but again, I would say it's again, this is all explained in our documentation, and at the end you enter the value. So in that case, the drug name. And yeah, you have several filters that allow you to query very specifically the data into ChEMBL, and for the output you can get by default XML, but you can also, just by adding JSON at the end of the URL, you will get a JSON format, but it can also be a YAML, and if you are querying for a compound, you can get the compound image if you add the SVG extension. And just to finish, so if you are interested in accessing ChEMBL through in a programmatic way, we recommend you to use Python. So it can be directly in a Python script, but also we found that Jupyter Notebook is very convenient to do this, and you can use the library Pandas, and you can use directly the API, the web services, but we also have a Python package that contains all the different functions that I have enumerated, and that allow you to query the database with just an internet connection, which is very convenient. And that's it. So I pass the floor to AJ.
Yeah, great. Thank you. So next question is mainly for open targets, I think, is, are there any resources that can be used to identify druggable gene based on protein-protein interaction analysis?
Yeah, it's a good question. So Open Targets does integrate information about protein-protein interactions. So we have a variety of different sources that we use to show you these. There isn't, so you can answer this question, but you kind of have to do legwork yourself. So you'd have to go and find the interacting proteins and then see the information that we have on them. It's not super straightforward to go from your interacting protein to, is this druggable for this target, if that makes sense. So the information is there, but you have to do the legwork to actually get the answer that you're looking for.
Yeah, and just to add on Elena's, we are trying, because we know this is a limitation of resource, we are trying to get this part better, so that the users doesn't need to click many times to find this straightforward question. Yeah, this is on the roadmap. So that was a very pertinent question.
Very good. Thank you. Next question is again for open targets. Is there a way to look up the available target residues noted for my proteins of interest, which I may use for drug designing?
Yeah, so again, so this is information that we integrate as well in our protein annotation, so target annotation page. So we've integrated data from ProtVista and from like we have the AlphaFold structures. So you can look up this information in a target annotation page, for example.
All right, okay, thank you. And we have another, I think this was the last question which came in through registrations. And I think, Nicholas, you already answered part of it. It was about the programmatic access. So how to use Python to extract data from databases and create the ontologies instead of doing it manually. So Nicholas, I think you touched upon this question already, but is there any other comment from you or from Open Targets?
And just to stress that we have a Python library that allows you to access the ChEMBL data directly. So well, if you code in Python, you can use it directly. It's convenient.
Yeah, in our case, I don't know about curating the ontologies specifically, but you can definitely use Python to access our data. We actually have, if you check out our documentation, so platform-docs.opentargets.org, we have some sample scripts using Python for you to access the data, for example, using the API or using the data downloads. So I recommend you check those out. There's also been quite a few questions answered on our community page using Python. And that's always a good place to put questions. If you have something that's specific to open targets, I would recommend you post there because our team will check it out and try and help you if they can.
Great, thank you.
And to answer more on the ontology part, I'm not sure if it's going to answer the question, but we have other resources at the EBI that allows you to curate on ontology. I mean, to associate words to ontology. And on top of my head, we have OLS, which is an ontology lookup service, and we also have Zuma. Which I'm using quite a lot actually when I need to map terms to ontology. So yeah, I recommend to use that.
Yeah, I think we also have Go and GOA from EBI, which provide the ontology service. I mean, yeah. And just to add to that, I mean, in addition to all these questions, we also have tutorials based on ChEMBL resource and also for open targets.